Hello friends, in this video I'm going to take this topic of enzyme regulation, that is how enzymes are controlled, right? So this is a way of control, regulation of enzymes, and there are various ways. Let's discuss. First one is allostric covalent modulation of enzymes, compartmentalization of cells, rate of synthesis or degradation of enzymes, and synthesis of inactive zymogens. The first one is allostric. This we've already discussed in the previous videos. You can check it there. And allostric can be either a form of activation of the enzyme or inactivation of the enzyme. We have allostric inhibitors, allostric activators, right? Next is covalent. Most common covalent modification is phosphorylation and dephosphorylation. By this method, many enzymes are activated or inactivated. You know that in fed state, insulin hormone is there which will cause dephosphorylation and will activate all the anabolic enzymes. But in fasting state, the hormone is glucagon which will do phosphorylation and activate the enzymes of catabolic pathway which are occurring in case of fasting condition, right? Next is compartmentalization of the cell. That is, different enzymes are kept in different compartments of the cell, and this way also we can do the control of pathways or control of enzymatic activities. Let's see the example. You know, like fatty acid synthesis pathway. Fatty acid synthesis is anabolic pathway. Any anabolic pathway occurs in cytoplasm, and the starting material for fatty acid synthesis is acetyl-CoA, which is required in cytoplasm for fatty acid synthesis. But acetyl-CoA is formed in mitochondria. In mitochondria, link reaction occurs, that is, pyruvate forms acetyl-CoA. This acetyl-CoA has to go from mitochondria into cytoplasm so that fatty acid synthesis can occur. So this is a way of control, that if body wants fatty acid synthesis to occur, then body will allow acetyl-CoA to go from mitochondria into the cytoplasm. And if body does not want this fatty acid synthesis, then this acetyl-CoA is not allowed to go from mitochondria into the cytoplasm. Similarly, one more example we can take. In beta oxidation of fatty acid, beta oxidation of fatty acid is a catabolic pathway. Any catabolic pathway occurs in mitochondria, right? So in mitochondria, beta oxidation has to occur, right? But first we have to activate the fatty acid for beta oxidation, and activation of fatty acid, that is formation of acyl-CoA, that occurs in cytoplasm, so that this acyl-CoA has to enter into mitochondria for beta oxidation to occur. So again, it is a point of control for body, that if body wants beta oxidation, then only this acyl-CoA, fatty acid CoA, is allowed to go inside the mitochondria. And if body does not want beta oxidation, then this acyl-CoA is not allowed to go inside mitochondria, right? So we're done with first three types of ways of enzyme regulation. The fourth one is rate of synthesis or rate of degradation of enzymes. So rate of synthesis means synthesis of enzymes. You know, synthesis of enzyme occur at the level of gene. And there are two types of genes. One type is known as housekeeping or constitutive genes. Housekeeping, also known as constitutive genes. Housekeeping means always active. These genes are for those enzymes which are always required in a cell. For example, enzymes of TCA cycle are always required. So I can say that the genes for making the enzymes of TCA cycle, they all are housekeeping genes, because TCA cycle is a vital cycle. So all the enzymes should always be present in the cell at any point of time, right? And other genes are inducible genes, that is, whenever required, they can be induced. For example, genes for the enzymes of gluconeogenesis. We don't require gluconeogenesis in a normal situation. Normal situation means fed state, right? But if those enzymes of gluconeogenesis are required, let's say it is a fasting situation, at that time we require it. So at that time, what will happen, that those genes of gluconeogenesis enzymes, they will be induced, so that now those enzymes will be formed and we can use these enzymes for this pathway of gluconeogenesis. So we're done with rate of synthesis of enzyme, which occurs at the level of gene, and other is now rate of degradation of enzyme, which is also controlled. That if the enzyme is degraded, then that enzyme is not working. And if we want the enzyme should be working for a longer time, then that degradation of enzyme will be delayed, right? Now there are two ways by which enzymes can be degraded. One is a very specific way, that is ubiquitin proteasome pathway, which is a very specific, specialized pathway for targeting the protein and degrading those proteins. This pathway is very specific, requires ATP, it is a regulated pathway. It occurs in cytoplasm and nucleus. There is tagging of protein with ubiquitin, and ubiquitin is also itself a protein which is highly conserved during evolution, right? On the other hand, other pathway is very non-specific pathway which is occurring in lysosomes, does not require ATP. It is ATP independent lysosomal pathway, and you know the name of enzyme is acid hydrolase, which is there. And this acid hydrolase enzyme is also very non-specific enzyme which can break down many macromolecules, not only proteins, it can break many macromolecules. So we're done with fourth way of enzyme regulation also, rate of synthesis of enzyme which occur at the level of gene, and rate of degradation, for which we have done two pathways. One is ATP dependent, other is ATP independent. Now the last way of enzyme regulation is synthesis of enzyme in the form of inactive precursors first, and when required then that enzyme will be activated. For example, all the enzymes of pancreatic digestion enzymes which are there, which are released from the pancreas, those are mostly protein breaking enzymes which have to come in lumen of the intestine to break down the digestive proteins, right? Now if these protein breaking enzymes are active inside the pancreas only, then it will destroy the pancreatic tissue. So we don't want that. First, when they are synthesized in pancreas, they are in inactive zymogen forms, and then when they reach intestinal lumen, there they can be activated. So that is a control on the enzymatic activity, that where we want to activate the enzyme, where we want that this enzyme should be working, only in that area of the body it will be working. For example, chymotrypsinogen, trypsinogen are formed which can be activated to chymotrypsin and trypsin, right? This is it with this topic of enzyme regulation. Thanks for watching. Best wishes to you.